Very good evening, everyone. Please confirm if you all can hear me. Is my voice loud and clear? Are you all able to hear me? Okay. Thank you for confirming. Very good evening, everyone. Welcome to day 2 session of Brahmastra Series. So as you all know, the CSIR NET Life Science exam is approaching. So I'm sure it's very near. So I hope that you all are revising the concepts and also practicing questions. So if not, then you can utilize this 1 hour for practicing questions. So in this live session, I will be giving you certain questions. We will be revising the concepts based on certain topics, and based on those topics only, we will be solving certain questions. You can learn the strategy also of solving questions, especially Part C questions which are asked in the exam. So I hope you all have taken a pen and a paper, right? The copy. So let's get started. So in this session, I will be discussing questions from Unit 8, that is Inheritance Biology, subunit Gene Mapping Methods. Now, this is a very important subunit from exam point of view, or I should say from the Unit 8. This subunit is very important because every year questions do come from this subunit, that is gene mapping methods. Now, the question might come from linkage mapping, or it might come from physical mapping, including deletion mapping or somatic cell hybridization, or tetrad analysis, and even molecular markers. Although molecular markers topic is common with Unit 12 and Unit 13 also, so you can expect combination-based questions from here. But in general, these are the topics from where the questions are asked in CSIR NET Life Science exam. Now, today I will be discussing, we will be revising the concepts based on these topics, and also I will be giving you certain questions for practice which are based on these topics. And also, you will be learning the strategy of solving Part C questions which are asked from these topics. And even you can analyze your preparation also, right? So let's get started. So here comes the first question on your screen. I want you all to go through this question. I know it looks lengthy, but don't get scared. Okay, this is you all are here to learn, right? So I will be giving you certain tips. Okay, the strategies of solving such questions. So first, try it yourself. I'm sure you all have studied, because now we have very less time in the exam, right? It's very near. So go through this question, recall the concept, right? And try solving this question. So question is, two mutations were isolated in bacteriophage. One causing clear plague and other causing minute plagues. Now the genes are responsible for these two mutations are 9 centimorgan apart. The plagues with genotype, so the genotypes are given, C plus M minus and C minus M plus, were mixed to infect bacterial cells. The progeny plagues were collected, cultured, and plated on bacteria. The expected number of different types of plagues are shown below. So here the different expected numbers of different types of plagues are given. So you have to find that which one of the following options represents the combination of all correct statements. So I'm giving you few minutes to solve this question, and you can post your question, you can post your answer in the chat. Okay, so let's start. And if you have any more question, any other question, you can just let me know in the chat. Okay, please utilize this 1 hour. Biotechnica has specially every year, like, we come up with this Brahma series, and because we know that how much this exam is important, how this exam plays a very important role. CSIR NET Life Science exam plays a very important role in a it can help you build your career, right? It is a gateway for several reputed institutes, right? So utilize this 1 hour, and you can also analyze your preparation, right? So focus on the data which is given in the question. Okay, So this question is based on linkage and mapping of genes on chromosome. So how do you map genes on chromosomes? Okay, Bhaskar, you have answered option 1. Good. Really appreciate your response. Please don't hesitate, okay? You all are here to learn. So right now we are just practicing questions, and it's very important that you should know that whatever concepts you are thinking, whatever strategy you are using, whether you are um, thinking in right direction or not, you are recalling the right concept or not, and most importantly, like, are you able to figure out like how to solve this question, right? So don't hesitate. Whatever answer you are getting, please let me know in the chat. Okay. So it's already mentioned that two mutations were isolated, right? So one causing clear plague, other causing minute plague. So what do you think? And they are 9 centimorgan apart. So see, I'll just tell you what should be the strategy of solving such questions. So first of all, read the question carefully, okay? And like when you're practicing questions, like you can pin, like you can underline or you can note it separately. What all are the important points or the data given to you in the question, right? So the information given is that there are two mutations which were isolated in bacteriophages. One is causing clear plaques, and other is causing minute plaques, that is small plaques. So the gene responsible for these two mutations are 9 centimorgan apart. So distance between the genes are also mentioned, that the genes which are responsible for these mutation is 9 centimorgan, right? Now, it is mentioned that plagues with these two genotypes, they were mixed to infect bacterial cells, and the progeny plagues, they were collected, cultured, and plated on bacteria, right? So first of all, here we have to recall the concept of linkage and how we can map genes on chromosome, right? So basically, linked genes are what? Genes are said to be linked that are located on the same chromosome and are usually inherited together. Okay. So let's say this is a chromosome. So in chromosome, we know we have a centromere region and a short arm, that is the P arm, and the long arm, that is the Q arm. And genes are located on chromosome, right? So if the two genes are very closely located, let's say A and B gene are very closely located, and let's say this is the C gene. So if you see the distance between A and B and A and C, definitely A and B distance is less. So it is the concept is that the genes are said to be linked if they are always inherited together. They are closely located on chromosome and they are most frequently inherited together. But remember, the linkage is like when the linkage is happening, the crossing over will not happen, right? Because recombinance we get when crossing over is happening. So like here you can see in this given figure, if there is no crossing over happening, then the gametes formed will all be parental type, like they will not be recombinant, they are non-recombinants, right? But the linkage, complete linkage, like we say complete linkage when there is no crossing over happening, but complete linkage is rare. There is a concept of incomplete linkage also, where sometimes the crossing over event may happen. Now, if the crossing over event happens, so crossing over happens in prophage one phase of meiosis, right? So when the crossing over happens, then in that case, there will be like you will be getting parental gametes. You will be getting like some will be recombinant, some will be non-recombinants, right? And in case of incomplete linkage, parental or I should say non-recombinants are always more in comparison to the recombinants. So frequency of recombination, another concept is frequency of recombination measures the intensity of linkage. How much the genes are linked, that we can determine by calculating frequency of recombination. So if there is a very tight linkage means case that when there is no crossing over happens, no recombination happens, then in that case, you have to learn this concept that in case of very tight linkage, the recombination frequency will be close to 0, right? Because no crossing over will happen. And in absence of linkage, the maximum recombination frequency that can happen is 50%, because half of the gametes form will be non-recombinant and half will be recombinant. And also there is a formula using which we can calculate the recombination frequency, and that is number of recombinant progeny by total number of progeny into 100. So these are the few concepts that you have to, you need to be thorough with or need to be familiar with for solving such type of questions, right? Now coming to the question. So Bhaskar, I can see you have changed your answer, and that is option 3. So let's check. Let's check out. So as mentioned that in the question, so right now I'm writing whatever is given in the question. Okay, so there is a cross given between the individual, like these two genotypes, right? And what else information is given? That genes are 9 centimorgan apart, right? So if you remember, I told you closer the two genes, less chances of them to undergo recombination, and this recombination percentage tells us about the intensity of linkage. So it's only 9 centimorgan apart. That means the genes are linked. So the proportion of when the genes are linked. So here I'm talking about incomplete linkage, okay? Because in complete linkage, no recombinants will be there. But in incomplete linkage, definitely parental types will be much higher. The gametes formed will be formed where the proportion of parental types will be much larger than the recombinant types. So here you can see this is the genotype of the parents, right? Parental types. And recombinant types genotype is given over here. So you can see the difference, okay? The parental types are like C plus M minus and C minus M plus, whereas the recombinants are C plus M plus and C minus M minus. So this genotype is only possible when the crossing over event is happening. Then, as I told you, distance between genes is recombination frequency percentage, and 9 centimorgan distance means recombination frequency is 9%, right? Now, what we are going to do is we will calculate the proportion of each recombinant. Okay. So since we are getting two type of recombinants, right? One is C plus M plus, another is C minus M minus, and frequency is 9%, which we can write 9% is 9 by 100. So I can write 0.09. So each recombinant proportion will be 0.09 by 2. That gives you 0.045. Similarly, we can calculate the proportion of each parentals, that is C plus M minus and C minus M plus. See, this is all information is given in the question itself. What I am doing is I'm just calculating the proportion based on the information given in the question. Now, proportion of each parental, you can calculate 1 minus 0.09 by 2. That when you solve, you'll get 0.91 by 2, and on solving, you'll get 0.455. Okay. Now, we will now analyze the data given. Okay, these 4 statements which are given A, B, C, and D. So if you see the data, okay, like in A, B, and C, like the values given are like 455, 45, 45, right? These are the values, except D. In all three, these values are given. So when you total them all, you will get 1000, right? Now you can calculate number of parental and recombinants. So basically, just now we have calculated proportion of each recombinant. So utilizing that, we can calculate number of each recombinants. That is, we have to find out, right, like which statement is correct, like whether this value given is correct for the recombinant or this value given for parental is correct or not, that we have to determine. So because the total progeny is 1000, so 0.05 into 1000, you will get 45. So that means recombinant should be 45. Similarly, we can calculate for parentals. So for parentals, it is 455. Now what you have to do is, we know C plus M plus is recombinant, not parental. So it should be how much? 45? Because parental should be 455. Now in A and B, you can see that the recombinants are given 455. So this is incorrect, right? Whatever values we are getting, it doesn't match with the data which is given for statement A and B, right? Similarly, for recombinants, it is mentioned for 45, C plus M minus, right? And here also, like you can see that if you look here, like for C plus M plus, which is recombinant, and C minus M minus, right? So these two values are not correct, right? C plus M plus recombinant should never be, like, according to the values we are calculating, it should be 45. So answer cannot be A and B. So A and B are incorrect. But if you check C, okay, if you now check the C statement, it is C plus M plus that are recombinant and C minus M minus 45. So yes, it is correct. And these are parental, they are 455. So it is correct. So definitely C is correct, right? Now, that means if I say C is correct, that means answer cannot be option 1 and 2. Now, it will be either 3 or 4 because we have to check now D also. Now, for D, the values given are different. So, again, here we will calculate, we'll add them all. Okay, 65, 680, 685, and 70. So, you'll get 1500. And again, we will calculate number of each recombinants, and also we'll calculate number of each parentals. So, these are the values that you will get. So, recombinants 0.045 into 1500, that gives you 67.5. Now, if you see here, C plus M plus in D statement and C minus M minus, that is both the recombinants, the values are ranging like 65 to 70. And we are getting 67.5. So, yes, it is possible. Similarly, if we look for parental, so C plus M minus 680, C minus M plus 685. And what we have calculated, we have got 682. So, yes, this is within the range. So, D can also be possible. And when I say D is also possible, that means answer is option 4 here. So, Bhaskar, just have a look where you have committed a mistake. I hope now it is clear why the answer is option 4 and not option 3, right? So, see, we need to, like, you have to go step by step. Okay, read the question carefully, and also you need to focus on these keywords, like whether you have to choose the correct statement or incorrect statement. Now it is mentioned you have to choose the combination of all correct statements, so you cannot say that C is only correct, right? Because D is also possible. So that is why answer here will be option D. Okay, I think you missed all correct statements, right? It should include all. And kindly note, everybody, we are starting with the new batch, that is Laksh batch from 10th January. So if you want to master the subjects which are included in the syllabus of CSI Net Life Science exam, or if you want to crack the CSI Net Life Science exam with good rank, you can definitely join our upcoming batch. And these are the features of this batch. You can go through it. Okay. And also you will be getting the MBTI, that is molecular biology techniques hands-on training and internship program, free with this, along with this batch. So if you want to join, you can always contact on our toll-free number. Okay. So any questions so far? All clear? So shall we discuss another question? Yes, are you all excited to solve more questions? Can I get some comments in the chat? Share a thumbs up emoji if you all are ready. Okay, great. So this is the next question. The following figure represents a physical map and a genetic map for 5 different genes A to E. Okay. Which one of the following statements based on the above is correct? So 4 statements are given. The region between B and C is more recombinogenic than the other loci. Second, in comparison to the region between A and B, the region between D and E is more recombinogenic. Third, 1 centimorgan is equal to 1 kb. And fourth, if more markers were mapped between D and E, the genetic distance between D and E is likely to decrease. Now, to answer this question, you need to be familiar with the concept of physical map and genetic map. So try solving this question, and kindly note, if you will like attend this session till end, you will be, there is a surprise for you all. Okay, so please stay with me. It's a 1 hour session only, right? And I have common, like, when I usually interact with the students, and here at Biotechnic also we receive lot of queries, and many students say that, ma'am, we don't get time to solve the question. So why don't you utilize this time, right? You don't have to search the questions here and there. We are only providing you the questions in the class itself, right? In this live session itself. So don't forget, like every day you will have a new session, okay? From different units, uh, we will be different experts will be coming up with different questions from different units of CSNet Life Science syllabus, okay? So you can utilize this 1 hour every day. The Brahma series session will be 6 to 7. So make sure that you attend. You don't forget to attend, okay? So within the class itself, like you can join the session and you can solve the questions, right? And even if you have any doubt, you can in the class itself, you can get your query being answered, right? Okay, Bhaskar, you have already answered. It's option 2. What about others? I'm not getting, others are not responding. Are you not practicing? See, you have to write and practice. Verbally, if you are solving, then like in the initial stage when you are practicing questions, it has to be like you have to write and practice, okay? Because there are chances you can commit mistake if you are solving it verbally, right? But this question is very easy. You can do it verbally also. But for the exam point of view, it's better that you write and practice. That's the good strategy. So for this question, you should know the difference between physical map and genetic map. So it's a very easy concept. So the concept here is that, see, what are genetic maps? When we talk about map tells us about the location, right? Like when you talk about the geographical map, so they tell you about the location of a place, right? Similarly, genetic maps, they provide you information about the arrangement of the genes along the chromosome, right? Like if this is the chromosome, so whether it's a gene present here or B gene over here or C gene, or is it a C gene or A gene or B gene, right? So arrangement, their order, like whether we have ABC, CAB, BCA, okay, like that. Order of the genes, arrangement of the genes, and the genetic distance between them. Like if you see these two are very closely located, right, in comparison to these two. So you can the genetic differences, distances also you can determine. And remember that genetic maps are constructed based on recombination frequency. So I told you the formula, right? Number of recombinants by total progeny into 100. And always the distances on genetic maps, they are measured in centimorgans or map unit. And remember, one map unit is always equal to 1% recombination. Okay. On the other hand, if you see physical map, so this is the genetic map where you can see the distance between the genes are measured in terms of centimorgan, okay, or map units. And they're based on recombination frequencies. They're constructed based on recombination frequencies. On the other hand, physical maps, they provide you the detail of actual physical distance between the genetic markers. Okay, so actually physical maps are considered more precise, more authentic than genetic map, because here the actual physical distance between the genes, the markers, they are determined. And remember that here the base pairs are used as a unit of measurement. Okay, so bases are used as a base pairs are used as a unit of measurement. So this is a concept that you have to recall while solving this question. Okay, now coming to the question, we'll go through each option one by one, because this is the strategy of solving such question. So very first statement is that the region between B and C is more recombinogenic than other loci. Okay. Now, if you see B and C, okay. Now here it is given, like I'm sure if you see the units, you can understand that here we are talking about the physical map, right? Because the distances is measured in base pair, and here in centimorgan, the distances are measured in genetic map, the distances are measured between the genes in centimorgan, right? So as the option, as the statement is about recombinogenic, that is, according to this option, the region between B and C is more recombinogenic, that is, more chances to undergo recombination than other loci. So since we are talking about recombination, we will look the genetic map. And you can see that the distance between B and C is given only 2 centimorgan, right? So 2 centimorgan means 2% or 2 map unit or 2% recombination only, which is not too very much, right? So if you remember that recombination percentage tells you about intensity of linkage. So closer the genes, less chances of them to undergo recombination. So although by looking at this figure, like this image, you might feel that, okay, there's too much distance, but you don't have to see this, okay? This is just to confuse you. You have to look at these values, the numbers which are given, right? The distance to centimorgan. So this is not correct. Then the option 2 is in comparison to the region between A and B, the region between D and E is more recombinogenic. Now, if we see A and B, that is, again, we are talking about recombination. So we'll look at the genetic map. So A and B distance is 9 centimorgan, but D and E is 20. Okay, again, don't see this. Okay, don't see this image. Otherwise, you'll get confused. You have to look at these values, 9 and 20. So obviously 20 centimorgan means 20% recombination, which is more than 9%, right? So yes, the region between D and E is more recombinogenic. This is correct. Now, option 3 is 1 centimorgan is equals to 1 kb. So kindly note that it's not correct. 1 centimorgan is equals to 1 million base pairs in humans, okay? Um, Average, not 1 kb. So this is again incorrect. Then fourth is if more markers were mapped between DNA, like if you place more markers between DNA, the genetic distance between DNA is likely to decrease. No, this is incorrect. It should increase, right? If you are placing, when more markers, they are mapped between the like DNA points as mentioned over here, definitely the distance will increase, right? Because additional markers will provide more detailed information, right? More detailed genetic, like they will provide more information about genetic recombination events that has occurred between the 2 points, right? So that's why the correct answer here is option 2, because option 2 is, the statement 2 is only correct. So Bhaskar, very good. Your answer is absolutely right. So this is what I have mentioned over here regarding the option 2. You can go through it. So well done, Bhaskar. Now let's solve third question. Now I'm sure looking at the image, you can easily determine that it is based on tetrad analysis. So the question is, the figure summarizes the observation following a cross between two haploid strains of Neurospora crassa having alleles. So two alleles are given over here. Okay. So there is a cross between the two strains, right? So obviously the nucleus, now the embryo form will be heterozygous, right? So the following statements were made. So fusion of these two will results into, these two cells results into the nucleus that is heterozygous, right? The following statements were made in one segregation, that is, here you can see the numbers are given in Roman. So one, segregation of alleles occurred in anaphase 2. B, crossing over between the centromere and the gene occurred in 20% of the myocytes. C, with reference to the two non-recombinant parental chromosomes, there are 6 different ways by which they can orient themselves at the equatorial plate. And D, the gene is 20 centimorgan away from the centromere. So which one of the following options represents a combination of all correct statements? So please try solving this question. So I'm giving you some time to solve it. Okay. So you can all solve this question. Again, here you have to recall about the tetrad analysis, and the hint is Neurospora crassa. So actually, when you study about tetrad analysis, there is a concept of ordered and unordered, right? So there are certain organisms, right, which produce an ordered tetrad, right? They have ordered tetrad arrangement, some have unordered. So just recall what kind of arrangement do we have in Neurospora crassa, and then like you have to recall further concepts about the ratio of the ascospores based on the arrangement. What do we get? So all that information you have to recall here. So I want everyone to solve this question, recall those concepts. Okay, I have given you already a hint. So those who are attending live session, please of Brahma series, please inform your friends and others who are attending, who are preparing for CSNLET Life Science exam to attend these live sessions. Okay, we are specially Biotechnica, especially conducting this for you to make you all practice the Part C questions, especially because we get a lot of queries where students they say that, ma'am, we want to practice more Part C questions, right? So especially this Brahma series has been organized for you all, right? Conducted for you all. So utilize this 1 hour. Yes, please let me know in the chats. What do you think is the correct answer here? So you have to choose the correct statements. The options which represents a combination of all correct statements. See, I can directly tell you the answer, but this will not going to help you, right? At the end, at the time of exam, you only have to recall the concepts, right? You only have to solve the question, right? So this, when we talk about conceptual skills, analytical skills, you have to develop, and it all comes with practice. So practice as many questions as possible, because then only you are going to develop the analytical and conceptual skills, right? Which are needed to solve the Part C questions, especially of CSN at Life Science exam. And the best part of such questions, when some figure is given to you or data is given to you, is that you don't have to think out of the box, right? Already the data is in front of you, just need to learn how to analyze that data, right? So here already the figure is given where different octad arrangement is given, right? So let's recall the concept first. So Neurospora crassa always forms the, it's like, it's as I told you that there are certain organisms which you need to learn, which forms, like which organisms have a ordered arrangement, a tetrad or octad arrangement, which organisms have unordered. So Neurospora crassa always have a ordered tetrad or octad arrangement. Saccharomyces cerevisiae is one of the example of unordered tetrad arrangement, right? So here you have to remember two cases that so octad is where you have like 8 ascospores which are arranged in an ascus. So here you have to consider two cases, one when there is no crossing over and another when there is a crossing over happening, right? So here we are considering the arrangement of spores in the sac, and we can study them to know whether the segregation of alleles are happening in meiosis 1 or 2. So always remember, when there is no crossing over happening, the segregation of alleles happens in meiosis 1. So meiosis 1 segregation pattern is observed. Okay. And you can remember the ratio also. So ratio always formed is 4 is to 4. Okay. So here you can see like it is 4 is to 4. Now depending upon how is the arrangement of the alleles. So like either this arrangement or this arrangement. But the ratio will be 4 is to 4 in octad and 2 is to 2 in tetrad. So tetrad is where you have 4 ascospore, octad is where you have 8. However, when there is a crossing over event happening, okay, so crossing over always we consider between the allele A and with that of the centromere. So centromere we consider as a point just to determine the crossing over event happening. And always remember, when there is a crossing over, the alleles they segregate during meiosis 2. So meiosis 2 segregation pattern is obtained, okay, is observed. And ratio here you get is 2 is to 2 is to 2 is to 2 or 2 is to 4 is to 2, because see there are 4 strands. So depending on which strands are undergoing crossing over, okay, so crossing over happens between the non-sister chromatids of homologous chromosomes. So if the crossing over is happening between strand 1 and 4, or strand like as shown over here between strand 2 and 3, or between 1 and 4 or 2 and 4 or 1 and 3 or 2 and 3, so accordingly we can get different like either we'll get 2 is to 2 is to 2 is to 2 or 2 is to 4 is to 2 arrangement. So this ratio you have to remember. And when the ordered tetrad, for the ordered tetrad, the recombination frequency is calculated using this formula, half into M2 SK by total SK. Why half? Because half of the SK, like they are only recombinant, like because crossing over is happening, right? So only M2 segregation crossing over happens, and M2 segregation pattern is observed when there is a crossing over. So half of the ascospores we will be recombinant. Now we will consider, like, we'll recall what all information is given. So in the question, it is given that Neurospora crassa having alleles, two alleles already mentioned, one represented with uppercase A and other with lowercase a, they are crossed. So as I told you, heterozygous, like when these two different cells they fuse, so we'll get a heterozygous condition, and then this nucleus will further undergo divisions, right? Meiosis 1 followed by meiosis 2 and mitosis, right? So we'll get ascospores, 8 ascospores will be formed. Now we have to recall, like now we have to analyze the figure, okay? So given figure, if you see, like if you see the first one, here the arrangement, like what ratio we are getting. So as I told you, ratio and when the segregation is happening in meiosis 1 or 2, these two concepts are very important. So here the uppercase allele A is represented with this dashed circle, the shaded circle, and the without shaded one is representing the another allele. So here you can see the ratio is 2 is to 2 is to 2 is to 2. Okay, these two are of one type, these two another, these two another, these two another. So 2 is to 2 is to 2 is to 2 we are getting. And not only in 1, we are getting the same in 4 also, right? So 1 and 4, they will show 2 is to 2 is to 2 is to 2 ratio. Now, if you see second and third, here we are getting 2 is to 4 is to 2 ratio, right? So second and third, we are getting 2 is to 4 is to 2, okay? I have mentioned in the slide also. So these 2, these 4 and 2. So 2 is to 4 is to 2, right? So 2 is to 4 is to 2 ratio, when do we get? When there is a crossing over event between the locus A and centromere. I have already showed you in the previous slide. And what kind of segregation pattern is observed? Second division or meiosis 2, right? Now, if you see fifth and sixth, what ratio they are showing? 4 is to 4, right? 4 is to 4. So here we are getting 4 is to 4. And 4 is to 4 ratio we get when there is a no crossing over event, right? There is no crossing over event happening, right? So when there is no crossing over, you will all get parental combinations only, right? And first division segregation pattern is always observed. Now we have analyzed the figure, right? Now we will consider the options given. So first is, or the statements given. So statement A is that in first, that is this one, segregation of alleles occurred in NF phase 2. How do we know that the segregation of alleles occurred in NF phase 2 or that is meiosis 2? So we know that if there is a crossing over, then meiosis 2 segregation pattern is observed. And what is the ratio we are getting here? 2 is to 2 is to 2 is to 2, which is the ratio that we get when crossing over happens. Either 2 is to 2 is to 2 is to 2 or 2 is to 4 is to 2. So this statement is correct, right? Because definitely this ratio indicates that crossing over event has happened between locus and between the locus A and centromere, and the segregation therefore occurred in NF phase 2 or meiosis 2. Then B is crossing over between centromere and gene occurred in 20% of the myocyte. Now, if the crossing over occurred, right? So crossing over events, like if you see, we are getting like 2 is to 2 is to 2 in 1 and 4, and 2 is to 4 is to 2 in second and third, which indicates crossing over events. So we will consider these values 16, 14, 18, and 12. So these are the number of SK observed when the crossing over has occurred, right? So that's why we will consider, we'll add them all. So we'll get 60 and total SK is 300. So we will calculate the, like we have to calculate in how much percent of the myocytes the crossing over has occurred. So 60 by 300 into 100, you will get 20% of the myocytes. So recombination frequency, number of recombinants by total progeny into 100. So here total SK is 300. So into 100, you'll get 20% of the myocytes. So this statement is also correct. Okay. Then C is with reference to the two non-recombinant parental chromosome. What are the two non-recombinant parental chromosome? The fifth and sixth, they are representing the parental, right? Because no crossing over event is happening here, right? 4:4 ratio you get when there is no crossing over. So obviously parental combinations you will get. So it is mentioned that with reference to the two non-recombinant parental chromosome, there are 6 different ways by which they can orient themselves at the equatorial plate. Now I'll give you a trick here. If in any question they ask you about how many ways are possible in which the chromosomes can orient themselves in equatorial plate, there is a formula that you can use, that is 2 raised to power n, where n is the number of chromosome pairs. Okay. Now here we have only one, right? So 2 raised to power 1 is gives you 2 only, right? 2 raised to power 1 gives you 2 only. So there are 2 ways, only 2 ways, not 6. And based on these only we are getting this arrangement. You can see, like here this one indicates this allele arrangement, and here this one indicates this allele arrangement. But over here in 6th, you can see this one is indicating this arrangement, alleles over here, and this one with this allele over here. So 2 ways only, right? Even it is shown in the question also. That is what I have mentioned here. So statement C is incorrect. It's not correct. Okay. So there are 2 chromosome and orientation is independent. So 2 ways which are already showed in 5th and 6th, and not 6 by which the chromosomes can orient themselves at the equatorial plate. And the gene is 20 centimorgan away from the centromere. Now we can determine using recombination frequency, right? And recombination frequency is proportional to distance between the genes. Closer the 2 genes, less chances of them to undergo recombination. Farther the genes, more chances of them to undergo recombination. So here we will use the formula to calculate the distance, which is for the ordered tetrad analysis is half into M2 SK by total SK into 100. So M2 SK, that is 60, right? So half of 60 is 30. So 30 by total SK, that is 300. You will get 10%. That is, they should be 10 centimorgan apart, not 20 centimorgan, right? So when you add them all, you will get 60. So we have half 60, right, divided by total progeny into 100. So that gives you 30, and further you can solve, right? So it is 10, right? So statement D is also incorrect. And since we have to choose the combination with all correct statements, so we have A and B only, because C and D are incorrect. So answer here is A and B. So Bhaskar, it will be option 1 and not 3. Okay. So don't please make sure that you don't do the guesswork. Everybody, this is very important, because in CSI Net Life Science exam, we have negative marking also, right? So don't commit a mistake. Okay. If you're not sure, then don't attempt such questions. But you can see that these questions are not very difficult, right? They are not very difficult. You don't have to think out of the box. Yes, certain concepts you need to be familiar with. If you are familiar with those concepts, you can easily answer such questions. Okay, now coming to another concept, and that is deletion mapping, right? So how to, when you construct a physical map, different strategies you use. One is deletion mapping, another is somatic cell hybridization, and also we have certain other techniques which can be used, strategies which can be used. So here this question is based on deletion mapping. Please go through the question. So question is, mutants of bacteriophage that carry deletions can be used to rapidly locate mutational sites of newly obtained mutants. The mapping is based on whether wild type recombinants can be recovered when the deletion mutant and the novel mutant are brought together. So 4 independent deletions, 1 to 4 of a region, were used to map 4 novel mutations, that is A to D. Okay. So here you can see the deletions are given, and also like 4 novel mutations A to D are given. So results of mapping are summarized in the table, where plus denotes recovery of wild type recombinants and this minus indicates the inability to do so. Now, certain statements are also given that it was observed that out of 4 novel mutants, no revertant was observed for mutant A. And the second observation is that mutant B and C do not complement each other. Now, definitely you should know what is complementation to answer, to understand when we say that B and C do not complement each other. So what is the meaning of it? So for that, you should know what is complementation. And also you need to be familiar with that mutations, right? Like how you can check for whether the mutation can be reversed or can be like revertance can be observed or not. Now, following conclusions were made. A, the mutation A lies within the region of deletion 1. B, mutation can be ordered as ADBC. C, mutant A could be a deletion, and D, mutant B and C are located on two independent cistrons. So which one of the following options represents a combination of all correct statements? So cistrons, they are like referring to the genes only, okay? Structural genes basically. So answer this question. So are you finding the strategies helpful? Are you now confident enough that you will be able to, you can solve such questions? Like once you are familiar with the strategy, the concept, tell me, what do you think? Will you be able to solve such questions in the exam? Please let me know in the chat. Now, again, although this question appears to be very difficult, but believe me, it's not that difficult. Okay, it's very easy. Just you need to be familiar with the strategy of how to solve such question. Yes, quickly, can I get some answer? Okay, option 3. Fine. See, in the exam, you have to, like, time is also important, right? So you have to take care of the time also, the speed. So you have to work on your speed and accuracy both for CSI and Life Science exam, right? So first work on accuracy and then speed. Fine. So let's have a look how to solve this question. So basically, what is deletion mapping? Deletion mapping is a method, like as I told you, physical maps. They can be different strategies can be used to construct the physical map, and one is deletion mapping. So deletion mapping is a method for determining chromosomal location of a gene. Okay. Now here, like if you see, this is one of the experiment which is given over here, that how can you determine, okay, where the gene is located in the chromosome. So here is one experiment where an individual that is homozygous for recessive mutation in the gene of interest. So the mutant allele is denoted with this allele, and the wild type is denoted with this. So a mutation in the gene of interest is crossed with individual that is heterozygous for deletion. So if you look here, this is an individual, so it has a mutant allele, and this is another individual which is wild type because it has the wild type allele. But in one of the chromosome, the deletion is there. Okay, so you can see this portion is deleted where the gene is present. So that is only deleted. So always remember, when such individuals are crossed, then the progeny which is formed will be half of the progeny which will display the mutant phenotype and half will display the wild type. Okay, the reason is that because here the gene is present in the deletion, the gene of interest is in the deletion region. So this one here we are getting heterozygous condition, right? So we have the wild type allele, so it will be wild type. Individual will be display wild type characters. But here you can see that the gene is present in the deletion region, so it is not there. So we have only the mutant here, right? So half of the progeny will display mutant phenotype. But if the gene is present, the region of deletion is present somewhere else. Okay. So here you can see that deletion region is present over here. The gene itself is not in the deletion region. So when such individuals are crossed, you can see that all the progeny will display wild type. Okay, because the gene is not within the deletion region. So you can see that this one will be also heterozygous, this one will also be heterozygous. So they will have a wild type allele and therefore they will display the wild type characters, right? So wherever recombination occurs between two strains to produce a wild type gene, the point mutation cannot lie within the region of deletion. Obviously, wild type characters means that the product of the gene is present, right? Product is there, and product is because the gene is present, right? So that means gene is not within the deletion region. But if the recombination cannot produce wild type gene, as in this case, this is reasonable to conclude that point mutation and the deletions are found within the same stretch of DNA. Now, how we are going to link this concept in solving such question? So basically, in the question, certain statements are given, and the two statements are that A is mutation A lies within region of deletion, and mutation can be ordered as ADBC. Now, very simple trick here is that you just consider that both the table and the figure given. Okay, so here you can see, like if you analyze the table for A, okay, for A deletion 1, 2, 3, and 4. So for A, you can see minus, right? Right. So actually, I'll go back to the question again. Plus denotes recovery of wild type and minus inability to do so, right? And also we have, these are A to D are novel mutations. So keeping this point in mind, A, B, C, D. So A mutation, you can see that here, this is indicating that like for deletion 1, 2, 3, and 4. So all you need to do is you just have to consider both together, the table also and the deletion figure which is given. Okay. For B, so just have to match the pattern. Okay. Like if I just draw whatever is given in the table, if I just place it over here. Okay. So here I will get minus, 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 right? Here I will get minus, plus, minus, minus, minus. Here I will get plus, plus, minus, minus. And here I'll get plus, 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 minus. So this is indicating deletion. Okay. Minus is deletion, indicating deletion, right? The inability to, as I told you in the question, it is mentioned, right? So mutations, what is the order? So where do you see such pattern minus, 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 minus in A, right? So this is A. Where do you see such pattern plus, minus, 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 plus, minus, minus? So this is D. Then plus, plus, minus, minus, that means this is B, okay, because here we see this pattern. And for C, it is plus, 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 minus, so that means it should be C. So order ADBC, yes, this is correct. And you can see that for the A, like deletion, like if you see for B, C, and D, right, the mutants, we are getting like plus, plus, plus over here, but here minus. So that means the deletion, mutation A lies within the region of deletion 1, right? So both A and B are correct, right? So you have to just overlap the figure and table. You have to compare, right? Then mutant A could be a deletion. Second state, the C statement is mutant A could be a deletion. Now see here, you have to recall the concept of how you can determine like whether revertants can be generated or not, because one concept, like we have this observation being given whether revertant regarding the revertant. So here some points I have added regarding the same, that when you analyze the mutations, especially the novel ones, one way to determine the nature of mutation is by observing whether revertant can be generated or not. So revertant can occur through several mechanisms. So revertant means where original DNA can be restored, okay, or there is a second mutation which can compensate for the effect of the first. So as mentioned in that question, that out of 4 novel mutants, no revertant was observed. This is already given in the question. Okay, the observation, 2 observations were there if you remember. One was that no revertant was observed. So this indicates mutant A could be a deletion. And this conclusion is drawn because deletion, unlike other type of mutations like point mutation, removes a segment of DNA that cannot be restored. Deletion means what? Like if you have certain region and if I just delete this part, right, so this is already gone, right? It is completely removed, so it cannot be restored through simple mutational events, so no revertant can be formed, right? So that's why statement C is correct. And these mutant B and C are located on 2 independent histones. So since it is mentioned in the question that B and C do not complement each other, that means they are on the same gene, okay? because complementation occur only if the mutations are in different genes. So this is again a concept that you have to remember. So based on the analysis, we can say that answer is A, B, and C, because we have to choose all correct statements, combination of all correct statements. So this particular point is very important, okay, whether you have to choose the all correct or one correct, two incorrect, or vice versa, or incorrect statement, okay. So you have to pay attention to these details given in the question. Okay. Now, one more question I will quickly discuss. I will discuss about molecular markers, okay, because since only 5 minutes are left, so this will be the last question for of today. So we'll discuss a concept of molecular marker here. So given below are two figures X and Y representing molecular markers and their profiles in parental lines P1 and P2 and F1 progeny. The following statements describe the nature or probable identity of markers in the above figure. So A to D statements are given. So panel X represents codominant marker, panel X represents a dominant marker, while Y represents a codominant marker. C's panel X could be SSR, that is simple sequence repeat marker, while panel Y could be RAPD marker, that is random amplified polymorphic DNA. And D's panel Y represents a dominant marker. So which one of the following options represents a combination of all correct statements? Now, again, these type of questions are very scoring. Okay, see, subunit D is not only important from exam point of view, but it is very scoring subunit also. Very scoring. Okay, you can easily score 4 marks. All you need to do is recall the concept, right? You need to learn how to analyze the given data, the figures, the tables, right? So here, to solve this question, you need to be familiar with two things. One is you need to be familiar with the terminology, like what is a dominant marker, what is a codominant marker. Secondly, you need to know how to analyze, and also like you need to be familiar with different type of markers, what kind of SSR marker, what is RAPD, what is RFLP, what is AFLP. And also you need to know how to analyze this gel profile, which is given over here. Okay. So these two things are very important. Okay, Bhaskar, you have already answered option 3. You are saying very good, very good. So let's check out if your answer is correct or not. So Bhaskar, I really appreciate your response, and I'm sure you are learning something new today, right? Some new concepts, new strategies, new tricks. Yes? So let's first go through the concept. So we know that there are different type of markers. We have morphological markers. So morphological markers are which are indicators of phenotypic differences, right? Like differences in the pigmentation, color, right? Like here you can see these pictures. So looking at these two pictures, you can say that okay, these two are not same, right? There's a difference in pigmentation. Then we have biochemical markers, also cytological markers, also cytological markers where you can identify the variation in organism based on changes in chromosome number, chromosome number, and the structure, the shape, the structure basically. Deletion is if it is there, or insertion is there. And then biochemical markers where we use isozymes and proteins, enzymes, like for example, a amylase enzyme is used as a marker, biochemical marker for for acute pancreatitis. And then we have molecular markers where the DNA sequences, they have been used to determine the variation between organisms. We have different type of molecular markers, hybridization based markers, which are non-PCR based. So one example is RFLP, then PCR based, we have RAPD, AFLP, and many more are there. And then DNA sequence based markers are also there. Now here, what is the concept of codominant and dominant? Always remember, codominant markers are those which tells you about the uh, tells you about both, like helps you differentiate between homozygous and heterozygous. So they can tell you about both dominant and recessive allele. But dominant markers, as the name suggests, it is just tells you about dominant allele. Like if you see these two images, you will find that here, this is the parent one, this is parent two. So you can see this is one band indicating one allele, this is another band indicating another allele. And since the progeny inherit the genes from parents, so two bands are observed. But if the dominant marker is used, it will only tells you about the dominant allele. Like you can see for P2, we are not getting any band here. And also F1, obviously the gene would have been inherited from P2 also, but for that we are not getting allele from P2 also. But we are not getting any band over here. So if I don't show you the genotype, you will not understand, okay, like what is the difference between this and this, right? Because it looks same. You will not be able to differentiate between homozygous and heterozygotes. Talking about the RAPD and SSR markers, so RAPD stands for random amplified polymorphic DNA and SSR for simple sequence repeat. Both are PCR based markers, but the difference is RAPD, like here we use the random primers, but in SSR selective primers. And the important point to note here is that RAPD is a dominant marker, SSR is co-dominant. Now coming to the statements which are given, so you will analyze the given figure, and it is mentioned that panel X represents a co-dominant marker. So if you look carefully, this is panel X. So for P1 we are getting a band, for P2 also we are getting a band, and for F1 we are getting both the bands, right? So it is a co-dominant marker definitely, but for panel Y we are not getting, right? So definitely panel X, it represents a co-dominant marker, so A is correct. And then B statement is panel X represents a dominant marker. So this is incorrect because just now we have said that panel X represents a co-dominant marker and Y is dominant, right? So Y represents a dominant marker given in statement D. So we can say that D is correct, but B is incorrect. And last statement is that panel X could be SSR marker. So SSR is a co-dominant marker, right? So panel X represents co-dominance. So yes, it is correct. And panel Y could be RAPD marker. So this statement is also correct. So since we have to choose the correct statements, combination of all correct statements, answer here is A, C, and D. Okay. So this is all about the part C questions from subunit D, that is gene mapping methods of subunit D of unit 8, that is inheritance biology. Since if you remember in the beginning, I told you that in the end there is a surprise for you. So here on your screen you will find a link. Okay, so using this you can download. It's a free download. It's a link using which you can free download some more part C questions, practice questions from this subunit, that is unit 8D of CSN at Life Science exam. Okay, and also the explanations are also given so that you can check like whether the concept you are thinking in solving such question is correct or not, or whether your answer is correct or not. Okay, so practice questions are there with detailed explanation and answer, and the link is on your screen, and you can download it from there. So this is from my end. Thank you very much, and all the very best. Don't forget, tomorrow again we'll come up with some more new questions from another unit of CSNET Life Science exam, and the session will going to held 6 to 7 p.m. every day, Monday to Friday. Okay, thank you very much. Take care.